Hello everyone. Today we are discuss new chapter number 17 locomotion and movements. Start करते हैं. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in the unicellular organism like amoeba is a simple form of movement. Movement of cilia, flagella, and tentacles are shown by many organisms. Human beings can move limbs, jaw, eyelids, tongue, etc. Some of the movement result in change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, swimming are all some form of locomotory movements. Locomotory structure need to be different from those affecting other type of movements. For example, in paramecium, cilia helps in the movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacle for capturing its prey and also using them for locomotion. We use limbs for change in body posture and locomotion as well. The above observation suggests the movements and locomotion cannot be studied separately. The two may be linked by stating that also locomotions are movements, but all movements are not locomotion. Method of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climates, conditions, or the escape for enemies. Types of movement. Cells of the human body exhibit three main type of movements, namely amoeboid, ciliary, and muscular. Some specialized cells in our body, like macrophages and leukocytes in blood, exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of protoplasm. Cytoskeleton elements like microfilaments are also involved in amoeboid movement. Ciliary movements occurs in most of our internal tubular organs, which are lined by ciliated epithelium. The coordinated movements of cilia in the trachea help us in removing dust particle and some of the foreign substance inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movements. Movements of our limbs, jaw, tongue, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human beings and majority of multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural system. In this chapter, you will learn about the types of muscles, their structure, mechanism of their contraction, and transport aspects of the skeletal system. Muscles. You have studied in chapter number 8 that the cilia and flagella are the outgrowth of cell membrane. Flagellar movements help in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protozoans like euglena. Muscles is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscles. They have special properties like excitability, contractibility, extensibility, and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscles are identified: skeletal muscles, visceral muscles, and cardiac muscles. सबसे पहले skeletal muscles देखेंगे. Skeletal muscles are closely associated with skeletal components of body. They have a stripped appearance under the microscope and hence are called striated muscles. As their activities are under the voluntary control of nervous system, they are known as voluntary muscles. They are primarily involved in locomotory action and change of body postures. Second है visceral muscles. are located in the inner walls of hollow visceral organ of the body like the alimentary canal, reproductive tract, etc. They do not exhibit any striation and are smooth in appearance, hence they are called smooth muscles, non-striated muscles. Their activities are not under the voluntary control of nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of heart. Many cardiac muscles cell assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature, and as the nervous system does not control their activities directly. Let us examine skeletal muscles in detail to understand the structure and mechanism of contraction. Each organism skeletal muscles in our body is made for number of muscles bundles and fascicles held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contain a number of muscle fiber. Each muscle fiber is lined by plasma membrane called sarcolemma enclosing the sarcoplasm. Muscles fiber as a syncytium as the sarcoplasm contain many nuclei. The endoplasmic reticulum, that is sarcoplasmic reticulum of the muscles fiber, is storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of a large number of parallelly arranged filaments in sarcoplasm called myofilaments or myofibrils. Each myofibrils has alternate dark and light bands on it. A detailed study of myofibril has established that the stated appearance is due to distribution pattern of two important proteins, actin and myosin. The light band contain actin and is called I band or isotrope bands. Wherever the dark band contain A or anisotropic band contains myosin. See the diagram. This is fascicle muscles bundle and दिस इज स्मॉलेस्ट मसल फाइबर्स जो कि सराउंड रहता है सार्कोलेमा जिसको हम प्लाज्मा मेम्ब्रेन बोलते हैं मसल्स की और ये है अपनी ब्लड कैपिलरी. Both the protein actin and myosin are arranged as rod-like structure parallel to each other and also to the longitudinal axis of the myofibrils. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thick and thin filaments respectively. In the center of each eye band is an elastic fibers called Z line which bisects it. The thin filaments are firmly attached to the Z line. The thick filament is the A band are also held together in the middle of this band by the thin fibrous membranes called M line. The A and I bands are arranged alternately throughout the length of the myofibrils. The portion of the myofibrils between two successful Z lines is considered as the function unit of contraction and called a sarcomere. In a resting state, the edge of thin filaments on either side of thick filaments partially overlap the free ends of thick filaments, leaving the central part of the thick filaments. This central part of thick filament not overlapped by thin filaments is called H zone. जो थिक फिलामेंट्स है वो है अपना मायोसिन और जो थिन फिलामेंट्स है वो है अपनी एक्टिन. ये है थिक फिलामेंट्स, दिस इज़ अ मायोसिन और ये है थिन फिलामेंट, दिस इज़ अ एक्टिन जो कि जेड लाइन से कनेक्ट रहती है. इसके बीच में जो एच जोन है जहाँ ये ओवरलैप नहीं करती है एक्टिन और मायोसिन एक दूसरे के ऊपर, और मायोसिन जहाँ एक्टिन जहाँ होती है वो आई बैंड बनाती है और जहाँ मायोसिन होती है वो ए बैंड होता है. ये जो पूरी यूनिट है इसको हम सार्कोमेयर बोलते हैं. दो जेड लाइन के बीच का पोर्शन, दो जेड लाइन, ये एक जेड लाइन ये से इनके बीच का जो पोर्शन है जहाँ एक्टिन है, मायोसिन है, एच जोन है, एम लाइन है, ये सभी सार्कोमेयर बनती है.